can observe the crossing over occurs. Okay. Then next you have diplotin is the diakinesis. So significance of the crossing over, actually why this crossing over is required? Here it is the molecular basis of genetic variation. And you can analyze the gene linkage by using this crossing over. So that is
These
why
meiosis steps are same of human being.
which one?
These pracatin, diplotin, these are same, no, ma'am.
Yeah, these are same. Almost they
Just
are
we have
all,
to
yeah.
figure out.
Prophase, see, actually, metaphase, sorry, in prophase you have 5 different stages, that is prophase, 4 different stages, that is prophase, metaphase, anaphase, and telophase. But this meiosis has actually, there are 2 types of meiotic division, meiosis 1 and meiosis 2. In meiosis 1, again it is divided into 4, that is prophase 1, prophase 2, prophase 3, and prophase 4 we say. Now, but that prophase actually is divided into these divisions, that is leptotin, zygotin, packaging, diplotin, and diakinesis. That is was it, not in what is that, mitosis. This is actually seen only in the meiosis. Okay, so it is almost the same. The different stages of cell division, whether it is a human being or whether it is an animal, with few exceptions it will be. Okay, but the rest of the protocol will be the same. Okay. So
Okay.
this is okay. Now see here again, the study of linkages between many genes enables us to create a linkage map. So if we know the linkages, okay, then we can know the linkages of many genes, because you all know that the genes are linked with each other. And if we study the linkages between many genes, it will help us to create a linkage map or a genetic map. So that is why this is very, very important. So what is genetic map? It is nothing but an arrangement of genes on the chromosome. Okay. So same way, Stuti went also postulated the same message he has given. He also stated that genes are arranged in a linear series on the chromosome. Okay, genes which are close together will be separated by crossing over, and by determining the frequencies of the crossing overs to plot the no, it's not required. We can say these are the two, that is they are arranged in a linear order, and the genes which are placed very close to each other will be separated by the crossing over. And we can calculate the frequency of crossing over by applying this formula. Okay. Then they are given. Now, just now we have talked about the gene maps, no, genetic maps, and the distance between any 2 genes actually can be measured in terms of the map unit. Now, MU. So, 1 map unit will be equal to 1 centimorgan, that is cm. Since it was developed or given by Morgan, so that is why it is called as the centimorgan. And next is the about the physical maps. What are physical maps? Now, physical maps are nothing but identification of the gene location on a chromosome and determination of the distance between the genes in base space. So the maps composed by direct DNA studying are called as the physical maps. Now, basically speaking, there are 2 types of maps. One is called genetic map and the physical map. So physical maps are what? Maps, when you talk about the maps which relate with the direct DNA, studying of DNA, then you say physical maps. Whereas genetic maps are nothing but these genetic maps, when you are studying about with reference to the DNA base pair and all those things, then you say call it as a genetic map. Now, physical map and the genetic map are almost similar, but they are non-identical. They are almost similar, but they are non-identical. Okay. Now, linkage mapping by recombination in humans. This is not required. This we have to talk with reference to only fishes. Okay. Next, we'll go to another one. Now, coming to sex determination and the inheritance of sex-linked traits. Now, now, what do you mean by sex determination? Nothing but determination of sex. So, sex is a set of features, so which provide the recombination of the genetic material during the reproduction. So, sex is a hereditary determined trait. And for example, in humans have got how many? 23 pairs of chromosomes, no? So, likewise, 22 pairs of autosomes are present, and 1 pair of allosome or the sex chromosome is present. Now, sex chromosomes, but in case of fishes, because here we have to talk in terms of fishes, no? In fishes, sex chromosomes have not been identified. So, authentically, there is no authentic proof whether the sex chromosomes are present in fishes or not. So, there is a little bit ambiguity, but in humans and other animals, actually, the sex chromosomes they have identified. So that is why actually in humans, we talk about 23 pairs, that is 46, out of which 22 pairs are nothing but the autosomes or the somatic chromosomes we can see, and one pair is a sex chromosome or the allosome. Now here, this is then sex is determined. You all know actually what do you mean by homogametic and the heterogametic. So the female produce actually only one kind of gamete with only the X chromosome. So that is why we call it as homogametic. Whereas males, what do? They produce two kinds of gametes. 50% of them will be sperms contain X chromosome, and 50% of them will contain Y chromosome. So that is why it is called heterogametic. Now, you all know sex is determined at the moment of fertilization by the kind of sperm that fuses with an ovum. Okay, so then you have X chromosome and Y chromosome. You all know X chromosome actually is a chromosome, is a female chromosome, and the male's Y chromosome is a male chromosome. Okay, the next is the discovery of sex-linked traits. Now, the discovery of sex-linked traits actually Morgan
And
published. Yes.
how do we identify the fish, sex of the fish?
Yeah, I will come to that later. I'm talking in general now. Okay, so this is all about in fishes. That is why
Okay.
they're saying no. I'll give you an example when I talk about the fish. I told you no, in fishes the sex chromosomes have not been identified properly, and they say most of the fishes research has been carried out, and the sex chromosomes actually they have not given a clear cut proof or a clear cut evidence about the sex chromosomes in fishes. They have given, okay, no doubt, they said about 50 chromosomes are present in some fishes, 40 chromosomes or 45 chromosomes, like that they have given the number, they have mentioned, but they have not mentioned the about the sex chromosomes. Okay, one or two examples are there where they have identified, but still there is a no such proof or an evidence. Okay, this is in general I'm talking about. Later we'll come to the fish. Okay, now next discovery of the sex linked traits. Now in 1910 Morgan published his research in his article like sex limited inheritance in Drosophila. Okay, so now what he did a lot of research. So we will see what are the sex traits in humans actually are characterized into three different types. One is sex linked, sex limited, and sex influenced. Now sex linked traits are what? They are controlled by genes which are present on X and Y chromosome. So they are related with the sex, no? So they are controlled by the genes which are present either on the X chromosome or either on the Y chromosome. Now all genes of sex chromosomes are divided into three groups. Like, okay, they may be the genes present on the X chromosome, okay, as well as on the Y chromosome. Genes of non-homologous, okay, not necessary. We can just say that the sex-linked sex traits are of three types. One is sex-linked, sex-limited, and sex-influenced. Sex-linked traits are actually controlled by genes. They are present either on the X and Y chromosomes. Okay. The next is, now, what is sex-linked? Dominant or the recessive character and Y-linked pattern of inheritance. Now, this is actually sex-linked or X-linked. It is called dominant mode of inheritance. Now, X is actually a chromosome which is present in the female, whereas Y is a chromosome which is present in the male. What is this mode of inheritance say? They are saying X-linked dominant mode of inheritance. Now, this is actually this inheritance or this character is exhibited by only the females. So here the females are affected more often by this particular inheritance. And if a dominant gene on the X chromosome causes a characteristic to be manifested in the offspring, and when the father alone is the carrier of a defective gene associated with the disease or disorder, he too will have the disorder. So his children will inherit the disorder as follows. Okay, so what we can say over here is, now we can say that X-linked, dominant or recessive and Y-linked patterns of inheritance are of come under three categories. One is X-linked dominant mode of inheritance. Then in this they have given, in this particular case, the females are more affected. Then second is a dominant gene which is present on X chromosome causes a characteristic that should be actually transferred to the offsprings. And if the father alone is a carrier of the defective gene, then he too will have the disorder. So his children actually will inherit the disorder like this. Okay. The next you have sex-linked recessive mode of inheritance. Now, sex-linked recessive mode of inheritance, actually, this disease usually occurs in the males. And these males who have inherited a recessive X-linked mutation from their mother. So here, what are the characteristic features? Is a male child of a woman who is a carrier has a 50% risk of inheriting the disorder. And a female child of a woman who is a carrier has a 50% risk of inheriting the gene mutation and thus being a carrier. Okay, so this is what they say. Another one is Y-linked mode of inheritance. Here in Y-linked, only men are affected. There is no question of female coming over here. Genes are simply passed from the father to all the sons, and they each affected son has an affected father. So these are the different modes of inheritance, that is sex-linked mode of inheritance. Okay, so this is all about. Now, actually, why I have given this introduction for you is, now you all know that in your syllabus you have sex-linked inheritance. So likewise, the what is that chromosomal theory of inheritance. Now, this is with reference to what is that human beings as well as with reference to other animals we have seen. But we have to talk with reference to the fishes. So in fishes, what are the different types of chromosomes? You all know the structure may be the same, okay, and the types may be the same. But okay, and also the chromosomal theory of inheritance also will be the same. But the thing is, how many types of chromosomes are present? It varies with from species to species. And what is the shape of the chromosome? And how actually, what are the various genetic manipulations, okay, that are being used for the gene manipulation and all those things. Now, this is only a basic introduction I have given you. I think yesterday no one has attended a class. Now, if you, how many of you are there? Only two of you are there, or only one? Hello? Hello, ma.
Yes, ma'am.
Only one is there. Okay. Then yesterday I talked about what is stereotype, what is karyogram, and all those things. Okay. Now we'll see tomorrow. You try to tell your friends to actually attend the class. Then tomorrow I'll talk about that. Now I will tell you about the what is that chromosome theory of inheritance is over. Now I'll talk about the transgenic fish production. Now in your syllabus, the fishes are also produced artificially, and the transgenic fish production, how actually the fishes are being produced using artificial method, and what are the various modern technologies actually that are being used. Okay, so wait a moment, I'll just share the material.
Ma'am, your voice is not audible.
Okay, now is it okay?
Yes. Yes,
Yeah.
ma'am.
Now, transgenic fish production. Now, what are transgenics? Transgenics means, or the transgenic animals are nothing but they are the organisms into which what we are trying to do, we are trying to put the transgene. Transgene is nothing but a recombinant gene has to be introduced, and this transgene or the recombinant gene, when it is integrated into their genome, into the genome. So, transfer of the transgene into the nucleus of a target cell, where integration into the whole genome will takes place. That is what we call as transgenics. Now, here you should have a basic knowledge about the recombinant DNA technology. You all know what is how to produce a recombinant DNA. Recombinant DNA is nothing but what we try to do is we have to isolate the DNA from one organism. Okay. After isolating the DNA from one organism, the gene of our interest, we have to take it, isolate it. After isolating, what we have to do, we have to, with the help of the vectors, we have to introduce into another organism. When we are introducing into another organism, what happens? Now this DNA and the DNA which is already present, they will combine and they form a new DNA. That new DNA we call it as nothing but a recombinant DNA or a chimeric DNA. That chimeric DNA or a recombinant DNA, or that is nothing but a transgene, that has to be again what we have to do, that has to be again transferred into another new organism. Okay, so in that new organism, again, what happens? This recombinant DNA, when we are introducing, okay, it will be totally the genetic constitution will be totally different, and it will not have, and when it develops into a new organism, it will not show the characters of their original what do you say, the organism. Okay, so that is what we call it as transgenics. So that is how the transgenic animals are being produced, whether it is any type of animal you take, whether it is fish or lion, sorry, what is that, chicken,
Sheep,
or
cow,
yeah,
goat,
goat, cow, whatever
pigs.
it may be. So the same technology actually is being applied. So that is what we call. So in the production of transgenics, how we are going to do that is the protocol is the same. Okay, the protocol is the same. So now a number of scientists actually have worked on it, and they are trying to produce the different transgenic fishes. Now you have seen Medaca is there, tilapia is there, okay, and a bass. Now different types of fishes actually have been produced artificially by applying this what do you say, gene manipulation techniques. Now the global scenario of the transgenic for other ornamental fishes. Now over the last 4 decades, actually various transgenic fishes actually have been developed all over the world, and these transgenic fishes actually expressing the growth hormone for aquaculture. So so these transgenic fishes that have been developed, they were used for aquaculture, industrial, as well as for pharmaceutical applications. And most of the transgenic fishes actually that were developed by using a growth hormone also had a highly conserved gene sequence. So these genetically modified fishes actually led to a dramatic development in the growth rate, thus showing the application of this transgenic in the aquaculture. So you all know actually why we have gone for these transgenics. Okay, because of the overpopulation and all those things, and for want of our selfishness, what we did is we tried to actually exploit not only the microorganisms, plants, as well as we have also exploited the animals, okay, for the human mankind or for the human welfare. Okay, so we try to produce the transgenic fishes like other animals, and these transgenic fishes actually, if you take it and we try to apply it in the aquaculture, you will get more amount of production, fish production. That means you can go about with more of fish production, which will be not only useful to the country, but also to the what do you say, the humans as such. So because fish is highly nutritious, you all know, and most of the people consume the fish because of that. So economically, as well as what do you say, the quality wise also, the fish serves as a one of the food, it is one of the food. So that is why the transgenic fish production actually led to the vast development. Okay, so it has a global scenario all over the world, not only in our country, but throughout the globe. Actually, the transgenic, what do you say, the fishes have gained a lot of significance. Now, this is about the next is what are the various methods. So apart from the food fishes, not only the fishes that are available and that we use daily, the ornamental transgenic fishes have also been developed using these transgenices using different color genes. So the value added aquarium fishes, now these days actually in aquarium, you see the ornamental fishes. They are not the regular fishes that we get it. So they are all ornamental fishes, and they are totally different colored. So the value added aquarium fishes also have already been commercialized, such as glowfish with 6 attractive fluorescent color combinations, including the starfire, red, cosmic blue, electric green, galactic purple, okay, sunburst orange, and moonrise pink. So like this, so not only transgenesis led to the development or the production of many, what do you say, fishes, but at the same time, it also helped in aquaculture for the production of the, what is that, ornamental fishes, you can say, or the value add fishes. These ornamental fishes, you all know, are different color. So how they try to produce is by using the color genes, they try to produce the ornamental fishes. Now, what are the various methods now in the production of transgenic fish? Now, coming to the different methods that have been adopted for the production of transgenic fish. Now, the first method is the micro-injection method. Now, in the micro-injection method, what they do is, now this micro-injection method actually has been used successfully in the production of fish, and it is a commonly used technique, and it is very simple and reliable technique. So the use of this technique actually results in higher survival rate for manipulating the fish embryos than the electroporation method. So it is very simple, it is reliable, it is very successful. Okay, and the most established method of for gene transfer in fish is microinjection. Now, microinjection, sorry, that allows the delivery of the transgene directly into the nucleus. So transgene is directly micro-injected into the male pronuclei of the fertilized eggs. So now see here. So here what they are doing is micro-injection. You need to take a syringe, and in that you have to take a transgene, and that transgene you have to directly inject it into the nucleus. So it is directly injected or micro-injected into the male pronuclei of the fertilized eggs. Okay, so that is how it is called as a micro-injection method. Micro means very small amount they take, use an injection, and they try to take a transgene, and that transgene they directly inject it into the male pronuclei of the fertilized eggs. Okay, so this is one of the method. Now, what are the disadvantages? Is it is time consuming and labor intensive. So here it takes a lot of time, and it is also labor intensive. A lot of laborers actually are required for this, and the nuclei of the fish eggs are small and they're difficult to visualize. Since the nuclei of the fish eggs are very, very small, it is very, very difficult to visualize. So that is why it serves as one of the disadvantage. Then the chorion hardens soon after fertilization, and in many fish, the pronuclei of the fertilized eggs is not visible, and therefore transgenes are usually injected into the egg cytoplasm. Since you are not able to, what do you say, the observe the pronuclei of the fertilized eggs, what do they do? What happens is the transgenes actually are usually injected into the egg cytoplasm instead of the pronuclei. So that is a major drawback. Then low efficiency of generation of transgenics. So with this technique, what happens is you get very little amount or low production of the transgenic fishes. So this is one of the method that is used for the production of transgenic fish. See here, they have given how they inject. Okay, so this is here, egg is immobilized by suction applied through the pipette. Now micro injection of DNA by using this, and here this is a suction applied through the pipette, and here this is the pipette for suction. So like this. Okay, now here you see the same way they have given. Okay. Next is what? Another disadvantage is to improve the efficiency of selection of the transgenic, genetic markers are co-injected with the transgene to monitor the transformed zygote. So the green fluorescent protein from jellyfish has been used for this purpose. Okay, so what they are saying is, though it has got disadvantages, no doubt, but still it is in use since it is very simple and it is reliable and also very easy to do. But only thing is, it is time consuming and lot of laborers actually need to be involved in this category. So to improve the efficiency of this particular type of what is a method, what they are doing is they are using the genetic markers, and these genetic markers they are injecting it with the transgene to monitor the transformed zygote. To monitor means after the zygote has been transformed, okay, what is its condition, how it is, whether it will further undergo the development or not. To monitor that, they are actually trying to inject the genetic markers. And for example, green fluorescent protein, that is GFP from the jellyfish, actually is used for this purpose in the zebrafish, that is transgenic zebrafish. Okay. Another technique that is used is called as the electroporation method. Now, electroporation method actually is chosen, and it is found to be the most effective means of gene transfer in fish, since a large number of fertilized eggs can be treated in a short time by this method. Okay. Whereas in case of the microinjection, you take only one or two eggs at a time. But in case of the electroporation method, what you can do is you can actually try to take many eggs at a time. So it will help you to transfer the genes. Okay, large number in the large number of the fertilized eggs. So within a short span of time, okay, this method can be done and involving a large number of the fertilized eggs. So it is easy to transfer the gene into a large number of fertilized eggs within a short period of time. So this is called electroporation. So here, electroporation, what is do is it utilizes a series of short electric pulses. Okay, it uses what? It uses a series of short electric pulses, means you have to use an electric current and give the electric pulses to permeate the cell membrane that make possible the formation of temporary pores on the surface of the target cells through which the transgene is introduced into the cytoplasm, where it is then delivered to the nucleus by the cellular machinery. Are you understanding here what they are saying? See, actually it involves what? It involves a electric pulse. So by the help of electric current, you have to actually generate an electric pulse. So this electric pulse, what it will try to do? It will try to permeate the cell membrane. You know what is cell membrane? Okay, the membrane which bounds the cell. And this cell membrane has to be permeable. Permeable in the sense means it has to allow, okay, it has to pass whatever actually that comes into way, it should permeate. So it should, the cell membrane should permeate the electric pulses. It should permeate the electric pulses means the cell membrane should be able to pass the electric current or to pass the electric pulses, okay, inside, which will help the formation of a temporary pores on the surface. So with the help of this electric pulses, what happens is when it is able to cross the cell membrane, then the temporary pores actually will be developed on the surface of the target cells. So when the temporary pores are developed on the surface of the target cells, through which what happens, you can easily introduce the transgene into the cytoplasm. Now, when once the transgene is introduced into the cytoplasm, from there it will be delivered to the nucleus by the cellular machinery. Are you understanding? So what does electroporation is? Electroporation actually involves a large number of fertilized eggs. So you can, what is that? Use, or you can transfer the transgene, okay, into a large number of fertilized eggs in a very short span of time. So, but what is a thing is it needs electricity. So it utilizes a series of short electric pulses. So when it is using a series of short electric pulses, that electric pulses or that electric waves has to pass the cell membrane. Okay, that means the cell membrane actually allow the electric pulses to enter into the nucleus, to enter into the cytoplasm. So what happens is this electric pulse, what it will try to do, it will try to permeate the cell membrane, which will make, when it permeates the cell membrane, the temporary pores actually will be formed onto the surface of the target cells. So when the temporary pores are formed on the surface of the target cells, when the electric pulses are passed, easily you can introduce the transgene, okay, into the cytoplasm, and from the cytoplasm it will be delivered or it will pass on to the nucleus with the other cellular machinery. So that is called as nothing but electroporation method. So electroporation method actually has been preferred in many laboratories for gene transfer in fish system because it of its efficiency, speed, and simplicity. So it is very efficient. It is, and within a short speed of time it will occur, and also it is very, very simple when compared to micro injection. Now the only drawback between this and that is in micro injection we may take only one or two cell at a time, or one or two fertilized eggs, but in electroporation large number of fertilized eggs actually can be taken, and we can we can transfer the genes into a large number of fertilized eggs in a short span of time, but there we cannot do it. So that is why electroporation method is preferred when compared to that. Now compared to disadvantages, see here how they are. Now electroporation cell process, so this is the cell, okay, this is the cell membrane. So you have, so introduction of genes or the drugs, then here electric field induces a voltage across the cell membrane. So when you are passing an electric current, okay, electric pulses given a shock, so it will become permeable. That means pores will be developed, I told you, no. So through these pores, what happens, the transgene can get easily introduced into the cytoplasm, and from the cytoplasm easily it can reach the nucleus. That is what, how it will take place. That is before pulse, during E field, that is electric field, and after pulse. Okay. Now disadvantages is that presence of a tough chorion layer around the fish eggs reduces efficiency. Removal of the chorion is a tedious procedure and introduces additional stress on newly fertilized eggs. So here what they are saying is actually the fish eggs consist of a chorion layer which is very, very tough. So to remove this chorion actually is a very tedious job. Extra additional stress is required. So therefore, it actually reduces the efficiency of this technique. Then the efficiency of the electroporation reduced because of voltage, number, and the frequency of pulses. So another disadvantage is now the efficiency of electroporation will be reduced because of voltage. Means the how much amount of current actually you are passing. So and also the number and also the frequency of pulses, means how many pulses, how many cycles, or how many frequencies of pulses, or number of what do you say, numbers you can say, or the frequency of the pulses you are giving, and how much of current or how much of voltage, the form of voltage or the form of ampere you are applying. So this matters a lot. So this also will reduce the efficiency of electroporation. So these are the two, I think, the drawbacks one has to follow. Then another one is also there, that is okay, and that is about the electroporation method. Now the third method is sperm mediated gene transfer method, that is SMGT. Sperm mediated gene transfer method means here it is a transgenic technique that transfers the genes based on the ability of the sperm cells to spontaneously bind to exogenous DNA and transport it into an oocyte during fertilization to produce genetically modified animals. So here sperm-mediated gene means, here what they are saying is the sperm should have the ability to bind to the exogenous DNA. The exogenous means the DNA which is present outside, and transport it into an oocyte during fertilization. So as a result, you can get genetically modified animals. So this is called sperm-mediated gene transfer method. So exogenous DNA refers to what? It is nothing but a DNA that originates outside the organism. So SMGAT. So this particular technique have low efficiency, and it is mainly due to low uptake of exogenous DNA by the spermatozoa. Okay, no doubt it will produce the genetically modified animals, but they are saying is exogenous DNA. Actually, I told you it is nothing but the DNA which is present outside the organism, and this particular method has low efficiency because the what is it? It low uptake of the exogenous DNA. So the sperms will not be able to take that much amount of DNA. So very low amount of DNA it can take up. So because of the low uptake of DNA by the sperms, this particular technique actually is not that what is useful when compared to the others. They are saying. Okay. The next is so mechanism. How this SMGT sperm mediated gene transfer method will take place. So here they have given now the exogenous DNA molecules first bind to the cell membrane of the head of the sperm cell. Then this binding and internalization of DNA is not a random event. Exogenous DNA interacts with the DNA binding protein that are present on the surface of the sperm cell. Then the spermatozoa are naturally protected against the intrusion of exogenous DNA by the inhibitory factor. And this factor blocks the binding of the sperm cells and exogenous DNA because in the presence of inhibitory factor, the DBPs lose their ability to bind to the exogenous DNA. In the absence of this, this DBPS on sperm cells are able to interact with DNA, and therefore they can then translocate the DNA into the cell. So therefore, the seminal fluid must be removed from the sperm samples by extensive washing immediately after ejaculation. So after the DNA is internalized, exogenous DNA must be integrated into the genome. That is what, though it looks simple, but it is a little bit complicated, where actually what we have to do is we have to see that the exogenous DNA is what? Nothing but the DNA which is present outside the organism. That DNA should bind to the cell membrane of the head of the sperm cell. Then the binding, the next is exogenous DNA. Actually, what it will do, it will interact with the DNA binding proteins that are present on the surface of the sperm cell. Then the spermatozoa actually are naturally protected against the intrusion of the exogenous DNA because the spermatozoa will not allow the exogenous DNA to get enter into it, and this is actually being affected by inhibitory factor which is present in seminal fluid. So this factor, what it will try to do, it will block the binding of the sperm cells and exogenous DNA. So because of that, it will not be able to enter into it. So it will lose its ability to bind to the exogenous DNA. So in the absence of this inhibitory factor, what we can do is the DBPs on the sperm cells are able to interact with DNA, and then they can translocate the DNA into the cell. Therefore, the seminal fluid must be removed from the sperm samples by extensive washing immediately after ejaculation, and after the once the DNA is internalized, means once the DNA enters into it, then the exogenous DNA also must be integrated into the genome. So that is how the spermatozoa. So this is DNA, then incubation, spermatozoa plus DNA, in vivo or in vitro fertilization you can go about, and then it will result in transgenic animal. Then the next one is retroviral injection method. Now, although transgenic techniques have been adopted successfully by microinjection and electroporation with great efficiency, but mosaicism is a major concern in resulting individuals. So therefore, a retroviral vector means such as long terminal repeat sequences of monovalent murine leukemia virus, and pantropic retroviral vector has been developed for improving the efficiency of integration of this gene. So because of its higher efficiency, it has been used to produce transgenic organisms, including the fishes. So therefore, now these viral vectors are what they are doing, they're inclined to unstable expression or even complete, silencing of the transgene expression and integration. So therefore, in a recent study, This pantropic retrieval vector has been used as a tool for transducing sea urchin embryos. And this is a valuable gene transfer technique for fishes in general, but introduction of viral sequences into the food fish may not be accepted by the public, but it could be used to ornamental fish development. Okay, so here they are saying what we are using a retroviral vector. Retroviral means viral vector. So though it has got efficiency when compared to the other gene, they are saying that it has been used to produce danzanic including features. Here, regrettably, these vectors actually they are unstable. They are actually inclined to unstable expression because these vectors cannot express the genes and they will not remain stable. They are unstable. So that is the reason why even complete silencing of the what is a transgenic expression integration rates may be increased because of active injection. So therefore, the successfully used retroviral vector or constants reported to produce antoptinos is the Pocillus lucida. So the vector that is used is nothing but pantropic retroviral vector, and it is a tool that is used for transducing sea urchin embryos. And this is a very valuable gene transfer technique in fishes, but the introduction of the viral sequences into the fish may not be accepted by the general public. So that is why they are using only for the what is it ornamental fish production. So here what you can see is this retroviral, just now I've talked about, no, retroviral injection infection method. So what you have to do is you have to actually inject a retrovirus. Okay, um, infect an retrovirus, what is it, the virus into the what is it, the sperm or the ova. So this what is it, the viral vectors that have been introduced, they are actually inclined to unstable expression. So they cannot express, the expression is unstable, and even complete silencing of the what is it, transgene expression integration rates are also increased because of the active infection. So that is the reason why, though it is being used, but it is used only for the ornamental fishes, but it is not used for the fishes. So it has got a drawback. So the disadvantage of this is what, the preparation of the retroviral particles, including the transgene of interest, is a very laborious process. It increases the cost and also it requires technology. So that is the reason why this particular technique, though it is used for the production of the transgenic fish, especially the ornamental fish, but for food fishes, that means the fishes which are edible, here they are not using this technique, they are not using that much. Okay. Now coming to the advantages of transgenic fish. Now, okay, we know now what are the various techniques that are adopted to produce a transgenic fish, and are these transgenic fishes useful? What are their advantages? We'll see now. So they say that transgenic fishes, the growth rate of transgenic fishes can be increased by 400% to 600%. Now they can reduce the feed input by up to 25% per unit of output, thereby improving the food conversion ratios. The next is transgenic fish have been developed for various applications. So transgenic fish are not only used for eating purpose, but also they are used as experimental models to study for biological research, environmental monitoring, for ornamental fish, and as well as for aquaculture production. Then 2 to 3-fold increase relative to non-transgenic fish have also been done for tilapia and Atlantic salmon. Then use of transgenic fish. Transgenic fish are also used as bioreactors for large-scale production of human therapeutic proteins or the novel foods. Then there are transgenic lines of tilapia engineered to produce human clotting factors. Then they are also used as models. It represents revolutionary advance. Then fish is as cost-effective and important animal models. They are used in genetics, developmental biology, as well as toxicology. And development of transgenic fish as a model in reducing or replacing the selected mammals used in toxicity testing. So what they are saying is, lastly, transgenic fish can be used in toxicity testing instead of the mammals that are being used. So you can replace the selected mammals, especially the mammals which are used as a models, or you can reduce the selected mammals and you can use the, in place of that, you can use a transgenic fish. So these are the different, what do you say, the advantages of the transgenic fish. Now we have seen the different methodologies or the techniques that are employed for the production of the transgenic fish. Hello? Yes, ma.
Yes, ma'am.
You're able to understand,
Yes.
no?
Yes, ma'am.
So these are actually the different techniques they have given. One is a microinjection method, the electroporation method, then you have the retroviral injection method, and you have that semen technique.